And welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Thursday, May 15th, 2014. The topic for today's webinar is immunotherapy for multiple myeloma. My name is Matt Tontinos, and I am a science writer here at the Cancer Research Institute. CRI is a nonprofit organization established in 1953 with the goal of advancing treatments that strengthen the immune system's power to conquer cancer. We fund scientists from around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an expert how immunotherapy is changing the face of cancer treatment. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your question at any time throughout the presentation by typing in the Q&A box on your screen. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website and on our YouTube channel. Now it's my pleasure to welcome today's expert speaker. Hearn Jay Cho is an assistant professor of medicine at the Mount Sinai School of Medicine and an attending physician with the Multiple Myeloma Service at Mount Sinai Tisch Cancer Institute. Dr. Cho's laboratory is investigating novel therapies targeting the type 1 melanoma antigen genes MAGE-C1 and MAGE-A3 in multiple myeloma. Dr. Cho is pioneering powerful new approaches to treat cancer with the immune system, and it's an honor he could join us today. As a reminder, we encourage you to type your questions into the Q&A box, and with that, I'd like to turn the presentation over to Dr. Cho.
Thank you, Matt, for that kind introduction. Good afternoon, ladies and gentlemen. Thank you for spending some time with us today to talk about immunotherapy for multiple myeloma. So I thought it would be useful for this discussion to give you first an overview of normal immune function, specifically talking about some of the important cells and proteins that play roles here, and discuss the specificity and memory of the immune system. And then go into the topic of cancer immunotherapy strategies that are being explored in multiple myeloma, specifically monoclonal antibodies, tumor vaccines, engineered T-cell therapy, and immune checkpoint inhibitors. So first, I'd like to introduce you to the cytotoxic T lymphocyte, also known as the killer T-cell. This is an important member of the immune system whose job it is to go around and examine cells in the body to try to detect evidence for infections and eliminate those cells that are infected. Like all cells in the body, the T cells express many different receptors, proteins that are designed to bind or recognize signals from the external environment. Probably the most important one is the T cell receptor, which is the way that T cells can examine other cells. The T cell receptor is sort of like a barcode scanner at the grocery store. It goes around and looks for signals from other cells. If it sees normal signals, then it passes by, and if it sees a signal that tells it that the cell is infected or the cell is abnormal, then it'll turn on the killing mechanism. There's a number of other types of receptors on T cells. There are activating co-receptors such as CD28. This is a receptor that amplifies or augments T cell function. And then there's inhibitory co-receptors such as CTLA4 or PD1. And these are receptors that put the brakes on the immune response from the T cell. It's a normal regulatory mechanism to turn off T cells when you don't want to have cell killing. And then T cells contain granules that have proteins that are effective in killing infected cells and other types of cells. That's sort of the payload for this type of immune cell. Another important cell is the natural killer cell, or NK cell. It also contains granules that have cytotoxic or killer activity. The natural killer cell has what's called an Fc receptor on the surface. This is a receptor that works in conjunction with antibodies, and I'll show you how that works a little bit later. Natural killer cells are an important member of the immune system that also plays a role, but it's not restricted by specificity to certain types of proteins, such as antigens, which I'll explain to you in a moment. The third player that I'd like to introduce you to is an antibody. Antibodies are part of your immune system. They're produced by B cells, and antibodies recognize a diverse array of proteins called antigens, which are related to infectious disease agents such as viruses and bacteria. And then there's also cancer-associated antigens that antibodies can recognize. Antibodies actually consist of two major parts. There's the antigen binding fragment, or FAB. Each antibody has two of them, and the blue portion represents the part of the protein that actually recognizes and sticks to the antigen. And there's also a constant fragment, which is this part of the protein. The constant fragment, or the FC portion, plays an important role on how the antibody signals to the rest of the immune system. Now, the important thing to remember is that T cells and antibodies represent what's called the adaptive immune system. And this is part of the immune system that can confer specificity, going directly for specific virus or cancer-associated proteins. And it also has memory. So there's long-lasting memory T cells and memory B cells, which remember the proteins or antigens that it's seen that it doesn't like. And if they're ever challenged again, such as if you get reinfected with a virus or if a cancer occurs, then they'll be able to rapidly mobilize an immune response to combat. So normally, T cells, such as killer T cells, are responsible for surveying the normal cells in the body. And they do so through a protein called the major histocompatibility complex, which is sort of a presenting system to show all the different proteins that are made inside the cell, the T cell. The T cell receptor can read proteins in the context of the major histocompatibility complex. So as I mentioned before, this is sort of like a barcode scanner. So the T cell receptor scans whatever protein is in the major histocompatibility complex. And if it's a protein that it recognizes as a self or normal protein, then that's a red light. So the T cell is not activated. There are certain conditions, however, where there may be some weak reactivity to normal self proteins. And that's where those inhibitory receptors come into play. So normal cells express what are called inhibitory receptor ligands, for example, PDL1, which bonds to PD1 on T cells. And that's an extra level of regulation that also can turn on the red light and prevent the T cell from being activated and killing the cell. So under normal circumstances, when there's a normal cell, normal proteins on the cell surface, T cells take a look at them, and if they're normal, they just pass them by. So the situation's a little bit different for when the cell gets infected. So for example, in this case, if a virus infects a normal cell, that virus starts to produce virus proteins, which are depicted by these gray boxes here. These are virus antigens, so they're not normal, they're not self. And those antigens go to the surface and are also presented on MHC class 1. And now, virus infections also downregulate those inhibitory surface receptors, such as PDL1, removing that break from the immune system. So now when the T cell scans the barcode on the MHC molecule, it gets a green light. This is an infected cell, and it should be activated. And so the granules are released, and it kills the infected cells. So that's normal T cell activity. Antibodies work in a similar fashion. So when a virus infects a cell, those virus-associated antigens can get expressed on the surface of cells, sometimes directly on the membranes. Antibodies that recognize, so these are non-self antigens. Antibodies that recognize those proteins will stick to the cell. Now remember, this is the Fc portion, and it communicates with the rest of the immune system. And so cells such as NK cells, which express the Fc receptor, can bind to the antibodies coating these infected cells, and that also activates the immune system to kill the infected cell. So that's normal immune function, and as I mentioned before, keep in mind that antibodies and T cells are specific for certain antigens. They're looking for abnormal proteins, such as virus-associated proteins, and they can also look for tumor-associated proteins as well. And it's important to remember that these immune mechanisms have memory and can confer long-lasting defense against these abnormal proteins. So cytotoxic T cells survey the self cells for non-self antigens. They kill infected cells. Antibodies stick to cells expressing non-self antigens and mark them for immune destruction. So we want to take advantage of these normal immune functions and direct them against cancer cells. And there's a couple of different ways that we can do this. So one strategy is targeted monoclonal antibodies. So antibodies that are designed to stick to tumor cells specifically and mark them for immune destruction. The second strategy is tumor vaccines, which can train T cells to recognize and kill tumor cells based on tumor-specific antigens. Third strategy is engineered T cells. We can directly engineer a T cell by adding a T cell receptor or antigen receptor to patient T cells so that they will go directly to tumor cells. And the fourth is checkpoint inhibitors. Now these are monoclonal antibodies, but they're, instead of being designed to mark tumor cells, they're designed to block the inhibitory receptor ligand signals that can put the red light or put the brakes on the immune system. So let's talk about monoclonal antibody therapy as it relates to myeloma. So myeloma cells express surface markers too, and one of them is CD38. And this is one of the surface markers that's being targeted by monoclonal antibodies. So if you engineer a monoclonal antibody that sticks to CD38, such as daratumumab, this is a monoclonal antibody that's in clinical investigation for myeloma right now. Daratumumab antibodies can stick to the CD38 on the surface of myeloma cells and recruit those NK cells through the Fc receptor, which will activate them, and then they can kill the myeloma cell, So this type of cell killing is called antibody-dependent cell-mediated cytotoxicity, or ADCC, and this is one of the major mechanisms for monoclonal antibodies to promote the killing of tumor cells. Tumor vaccines is a second, this is a strategy designed to train T cells to recognize and kill tumor cells such as myeloma cells. Here's an example of how this works in this cartoon. So myeloma cells, as I mentioned earlier, express myeloma-associated protein antigens. For example, MAGE-A3, which we study in my laboratory. MAGE-A3 is a protein that is somewhat restricted just to myeloma cells, so it's a good marker for myeloma cells that you would want to activate the immune system to. So we can prepare vaccines that are made with the MAGE-A3 protein antigen, and we can vaccinate patients with these vaccines. And after multiple vaccinations, we can stimulate T cell and B cell activity that is directed against MAGE-A3. So in the patient's bone marrow where the myeloma cells live, MAGE-A3 specific T cells that are generated by the vaccine can recognize MAGE-A3 presented on the surface of these myeloma cells, and then those T cells can kill myeloma cells. In addition, MAGE-A3 specific antibodies can bind up MAGE-A3 either in the environment or that's released from dying cells and help amplify the immune response through a variety of mechanisms. A variation on the strategy is engineered T cells. So in this strategy, blood is removed from patients and T cells are isolated from that blood. Now we can program these T cells to express a specific T cell receptor. So for example, as I showed you in the previous slide, you can generate T cells that recognize, for example, MAGE-A3 using vaccine strategies. Some patients will spontaneously generate immune responses against tumor antigens such as MAGE-A3. And it's possible to copy that gene for the T cell receptor out of those T cells. And that's this T cell receptor transgene, which is depicted here. And we can insert that gene into patient T cells so that the patient T cells will also express that myeloma-specific T cell receptor, for example, a MAGE-A3 T cell receptor. Then, in the laboratory, we can expand those T cells to generate millions and millions of T cells that are specifically directed against MAGE-A3 in the myeloma cells. And those can be reinfused back into the same patient, which is called adoptive T cell therapy. And those MAGE-A3 specific T cells can then go to the bone marrow and kill myeloma cells. And this is a strategy that's also under investigation in clinical trials for myeloma. So how does it work? So T cells that have been programmed either through a vaccine or through engineered T cell therapy to recognize myeloma-associated antigens will read that barcode on the surface of the myeloma cells and see the abnormal protein, such as MAGE-A3. And it gets the green light. So this is a signal that it can kill that the T cell can kill the myeloma cell. And in many cases, this may be sufficient, and clinical responses to T cell vaccines and engineered T cell therapies have been observed. But even in successful T cell therapy, there have been cases where even though there's a robust T cell activity, there aren't accompanying clinical responses. And it's likely that a lot of this is mediated by those inhibitory receptor ligand pairs, such as PD-1 and PD-L1. So PD-L1 expressed on the surface of myeloma cells will stick to the PD-1 on the surface of the T cells, and that sends up the red light. So it's putting the brakes on an immune response that is trying to kill the tumor cells. So how do we get around that? This is where the checkpoint inhibitors come in. So monoclonal antibodies that are designed to bind to either the PD-L1 or the PD-1 can interrupt this interaction and block it, thus removing the brakes from this immune response, and then releasing the T cell to kill the myeloma cells. So actually, the checkpoint inhibitors, such as the anti-PD-L1, are also now entering the clinical arena for investigation in multiple myeloma. So what are our challenges now? So for monoclonal antibody therapy, the challenge is finding the best myeloma-specific targets to design antibodies against. For tumor vaccines, finding the best myeloma antigens and vaccine formulations to stimulate strong T cell immunity. For engineered T cells, it's finding the best targets for T cell receptors, and for other types of receptors such as what are called chimeric antigen receptors, or CARs. And for the checkpoint inhibitors, what are the best inhibitors? Which targets should we be going after to remove the brakes from the immune system? And when should we give them? What are the best stages of disease to apply these types of therapies? And then finally, what are the best combinations of immune therapies? As you can see, all of these strategies are somewhat complementary, and it's easy to see how you could potentially combine some of these strategies in order to generate effective anti-tumor immune responses. And so the vision that we have for the future is that targeted therapies, including targeting antibodies, can be used to induce remission in patients with multiple myeloma. And then, using combinations of vaccines, engineered T cells, and immune checkpoint inhibitors, stimulate strong and durable anti-myeloma immunity to eliminate minimal residual disease after that initial therapy. And the advantages for this type of therapy is that it's specific. It should only go after myeloma cells that are expressing the abnormal proteins that we're targeting. And it should be durable because of the immune memory that we discussed earlier. So it's likely that current clinical trials for single strategies such as vaccines alone, engineered T cells alone, or checkpoint inhibitors alone will pave the way for combination strategy clinical trials. For example, using a combination of a vaccine and a checkpoint inhibitor, or an engineered T cell and a checkpoint inhibitor, or combinations with monoclonal antibodies directed against tumor-specific targets. And that's the end of my formal presentation now, and I'm happy to take your questions.
Okay, thank you very much, Dr. Cho, for that excellent talk. And so for the Q&A section, once again, if you have questions, just type them right in the box, and I'll try to get to as many as I can. As a first question, I wonder if you could talk about which of the 3 different therapeutic approaches that you've presented are furthest along in clinical development, and maybe sort of talk about how much further we have to go with each of them.
So the targeting antibodies have been developed the most in multiple myeloma. So right now, there's 2 monoclonal antibodies that target myeloma cells that are in clinical development. One of them is called daratumumab, as I mentioned earlier. That targets CD38 on the surface of myeloma cells. The second one is called elotuzumab, which targets another surface protein on myeloma cells called CS1. Elotuzumab is an interesting story because as a single agent, a single antibody treatment alone, it did not show a lot of clinical activity, but it was safe. And when it was combined with chemotherapeutic agents that are effective in myeloma, particularly the immunomodulatory drugs such as lenalidomide or proteasome inhibitors such as Velcade, they showed significant activity, and it was long-lasting activity. So there's a phase 3 trial for elotuzumab in combination with either Revlimid or Velcade in relapsed multiple myeloma, and there's plans for clinical trials for upfront treatment, including elotuzumab. Daratumumab is right behind it. That was the first monoclonal antibody to show single-agent activity in multiple myeloma. So it's rapidly moving through the clinical trial stages. I would expect we're going to see advanced phase 2 or phase 3 trials for daratumumab in multiple myeloma very soon. I think both of these agents are very promising, and we're hoping to replicate the success of, for example, Rituxan, which is commonly used for lymphoma. I think vaccines and engineered T cells are right behind these monoclonal antibodies. In addition to the clinical trials for recombinant MAGE-A3 that we're conducting with our co-investigators, there's a number of other vaccine strategies targeting MAGE-A3 and related antigens, such as cancer testis antigens. And there's a number of different vaccine platforms that are being tested. So, for example, we're testing a recombinant full-length protein vaccine in combination with immune-stimulating vaccine components. But there's other vaccines, such as dendritic cell-based vaccines. So these are cell-based vaccines made with the patient's own immune cells. And then there's a number of whole myeloma tumor cell vaccines that are being studied, such as dendritic cell myeloma cell fusion vaccines. There's several engineered T cell trials that have either are currently underway or recently completed, looking at T cells engineered to express T cell receptors specific for MAGE-A3 and other types of myeloma-specific proteins. The first clinical trials for checkpoint inhibitors in multiple myeloma are just getting underway now. So this year, a couple of clinical trials have opened, phase 1 clinical trials looking at anti-PD-1, anti-PD-L1, and anti-CTLA-4 in multiple myeloma. So everything's in the pipeline, as it were. As I mentioned, the targeting monoclonal antibodies are further along and are close to phase 3 trials that will hopefully lead to FDA approval in in multiple myeloma. We have several vaccine and engineered T cell therapies which are just behind. And then we expect that the checkpoint inhibitors will move quickly through the early stage clinical trials and hopefully lead to combinations, as I mentioned. I think that's really our goal, is to get combinations of all 4 of these strategies into clinical trials to effectively mobilize the immune system to fight multiple myeloma.
Okay, great. Thank you for that. And another question is, at what point should a patient consider going on immunotherapy? When they're first diagnosed, when their cancer comes back, or when they're in remission? Can you talk a little bit about when these come into play for a patient with multiple myeloma?
So that's a fair question. So just to put it into context, multiple myeloma is very different now than it was just 10 years ago, because there's been 2 new classes of chemotherapeutic agents, the IMiD drugs, the immunomodulatory drugs, as I mentioned, Revlimid, and proteasome inhibitors such as Velcade, which have revolutionized the care for multiple myeloma. So myeloma patients respond very well to these treatments, and they have longer remissions with these treatments. So 10 years ago, the median survival for this disease was around 3 to 4 years. And now, a rough estimate is that the median survival for patients with myeloma is at least 7 or 8 years. So we have more effective treatments and we have more durable treatments for myeloma. But unfortunately, today, 2014, we don't have a broadly applicable curative therapy, so that we're still looking for curative therapies for multiple myeloma. So for newly diagnosed patients, there's safe and effective therapy to put patients in remission. The question is, can we add something to that initial therapy? So for example, there's a phase 3 trial opening combining elotuzumab, the anti-CS1 antibody, with standard initial therapy for myeloma, such as Revlimid and Velcade. And if that trial is available to you as a patient, that's certainly an opportunity to participate in clinical investigation and advance our knowledge of the effectiveness of this type of treatment. But outside of a clinical trial for initial therapy, there's no immune therapy that we can necessarily add to standard therapy. By necessity, a lot of these treatment strategies, such as engineered T cells and checkpoint inhibitors, are starting out in the relapse setting. So for patients with multiple myeloma who've relapsed, there's a number of opportunities to participate in clinical trials for these new agents. And that's a real opportunity to benefit from this type of novel therapy.
Okay, great. this might be sort of hard to answer without talking about a specific patient, but how does a patient evaluate, say the patient has relapse myeloma and they're interested in going on a clinical trial for immunotherapy. How do they decide which of these avenues is right for them?
Uh, that's a tough question. I think the most important thing for a patient who is facing hard decisions about their treatment, I think the first step is to have a consultation with a myeloma specialist who uh, has clinical trials to offer to patients. Um, so it's it's important to get a perspective of what's available to individuals from a specialist, and uh, you can have a individualized discussion during these types of consultations. For example, there's a lot of variables that might play into a decision about whether you want to try an immunotherapy clinical trial or not. What kind of response you've had to previous therapy, how many previous therapies you've had, how fit is your immune system, if there's a reasonable expectation that the patient will benefit from an immune therapy. And so I think the consultation with a myeloma specialist is an important first step, preferably at an academic medical center where a number of different clinical trials are available, including immune therapy trials, because there are going to be cases where maybe an immune therapy is not necessarily the best choice for an individual patient. So having as many options available and a specialist's opinion and input available is very important for making those decisions for the individual patient.
Okay. Can you talk a little bit about the place of stem cell transplants in these immunotherapy trials? Is that often a component for trials, or
yeah,
can
it's
you?
definitely one of the areas where we're very interested in developing immunotherapy. So what Matt's alluding to is the standard treatment for most patients with multiple myeloma is divided into a couple of phases. So for a newly diagnosed patient, the first step is induction chemotherapy. So that's the initial therapy. The goal is to get patients into remission. And the backbone of these types of therapies are Velcade, Revlimid, and a steroid called dexamethasone. And we're fortunate that this approach is successful in the majority of patients. So we can put patients into remission with this initial therapy. But we know that there's still residual disease. So the standard second step is what's called consolidation therapy. And for the vast majority of myeloma patients, this is what's called an auto stem cell transplant. That's a procedure where a patient's own blood stem cells are removed and stored. Then the patient typically is admitted to the hospital and receives a very high dose of cytotoxic chemotherapy, so an older type of chemotherapy called melphalan. And that's a dose that's high enough to kill a lot of myeloma cells, whatever is left over, most of what's left over after the induction chemotherapy. But it's also very toxic to the bone marrow. So after the chemotherapy is washed out of the system, we give the patient their own stem cells back. And that's why it's called an auto transplant. It's not really a transplant. It's a method to support very high doses of chemotherapy. But the plus side of this procedure is we know that it extends disease-free and overall survival, and it confers upon the patient what's called the minimal residual disease state. That's the maximum elimination of bad tumor cells from the bone marrow, which is where myeloma cells live. So that's the least amount of tumor burden that a patient's going to have, and so it's probably our best opportunity to mobilize an immune response and eliminate those tumor cells. So how to augment the immune response against myeloma cells at a point where there's the least amount of tumor cells to eliminate, it's a very hot area of investigation. So for example, the MAGE-A3 vaccine trial that I conducted with my colleagues at Memorial and Fox Chase Cancer Center was in patients who were undergoing auto stem cell transplant. And similarly, a lot of vaccine and engineered T cell therapies are being investigated in this setting as well. I should say, though, that the whole rationale for investigating immunotherapy in multiple myeloma is grounded in the transplant experience, but in allogeneic transplant. So allogeneic transplant is a real transplant. This is when the host's immune system and myeloma cells are eliminated with chemotherapy, and those are replaced by blood stem cells from a donor. And allogeneic transplant is immune therapy. It is grafting donor's immune system into the patient. And in multiple myeloma, the only documented long-term cures have been in patients who've undergone allogeneic stem cell transplant. Now, unfortunately, allogeneic stem cell transplant is a very high-risk procedure, particularly for myeloma patients. And because of the average age of myeloma patients and because of the general state of health of most myeloma patients, this is not an option that we can use in the vast majority of myeloma patients. So it's a very high-risk procedure, but it's also the only procedure thus far that has documented long-term cures. So we want to replicate that allogeneic transplant experience by mobilizing immune responses in patients to eliminate those myeloma cells. So hopefully we can confer the benefit similar to allogeneic transplant without the risks.
Okay, great. Another question is, you alluded to clinical trials with checkpoint blockade antibodies beginning soon. Do we have any early clinical experience or even from animal models to have a sense of how effective we think checkpoint blockade antibodies are going to be in this form of disease? You know, given that in melanoma, for example, they seem to be very effective, and also maybe in lung cancer as well. Do you have any sense of where multiple myeloma is going to fall, or is it too early?
I think it's a little too early to, well, so we have a lot of hope because checkpoint inhibitors such as anti-CTLA-4 and anti-PD-1 have been incredibly successful in really bad diseases such as metastatic melanoma, lung cancer, etc. And it's important. We have a reasonable rationale to investigate them in myeloma because, for example, myeloma cells express high levels of PD-L1. So that's probably an important mechanism that myeloma cells use to escape the immune system. So if we can disable that mechanism, we can hopefully mount more effective immune responses against myeloma. There have been a couple of small trials looking at anti-PD-1 in multiple myeloma. The results haven't been reported yet. And now there's trials opening looking at anti-PD-L1 and combinations of anti-CTLA-4 and anti-PD-L1. They're open to all blood cancers, not just multiple myeloma, but multiple myeloma is one of the specific areas of focus within that group. And now those trials are open. But as I said, they're early phase, sort of phase 1 clinical trials. And there's a number of clinical trials specifically in myeloma that are in the planning stages that I would expect are all going to open within the next year or so. And that's looking at these are clinical trials featuring anti-CTLA-4, anti-PD-1, and anti-PD-L1, and various combinations of those agents. And There's several different antibodies from different companies for each of these targets. So it's important to find out from the clinical experience which are the safest and which ones are going to be effective in multiple myeloma.
Are the early trials going to start with combinations, or do you have to go first through individual like evaluation of the monotherapy of just a single agent before you combine them?
So it's not mandatory that combinations be tested individually first, but that's typically the way it goes. I heard that there is so so phase 1 clinical trials are primarily for safety to ensure that individual drugs or combinations of drugs are safe to give to cancer patients. And some of those combination trials have already happened in solid tumor, for example, in melanoma. Anti-CTLA-4 and anti-PD-1 have already been combined and found to be safe. And that paved the way for more advanced trials looking for clinical effectiveness. But that safety data can potentially be applied and as a rationale to go ahead and test combinations right off the bat in myeloma patients. So I'm aware of some clinical trials looking at combinations that are in the planning stages and hopefully will open soon. As I mentioned, some of the so-called basket trials. So these are clinical trials for a given agent that'll be open to, for example, all blood cancers. Not just myeloma, but lymphoma, leukemia as well. Some of the basket trials are featuring combinations right off the bat.
Are there certain institutions or centers that are especially focused on melanoma trials? And is there sort of a first place where people should go to look for trials?
Well, there's a number of institutions that have specialized services for multiple myeloma. So there's a concentration of clinical specialists who are running clinical trials, along with research scientists who are doing the translational and basic science to understand how these new agents work in multiple myeloma and hopefully design better, more effective, and ultimately curative treatment. There's a number of specialized myeloma centers in the U.S. An easy way to find them is to look. The Multiple Myeloma Research Foundation is a charitable organization that's very deeply involved in the development of new therapies for myeloma. They have a number of patient resources on their website that can direct patients to a specialty center in their geographic region. It just so happens in the New York area we're blessed with a number of very well-known clinical centers for multiple myeloma, not only our service here at Mount Sinai, but also at Cornell Weill Medical College, Memorial Sloan Kettering, Columbia, and Hackensack Medical Center. So some areas, there's a high concentration of specialty centers. Other areas, there may be one big referral center for a region. An easy way to find them is through the Multiple Myeloma Research Foundation. Another easy way is to look for where clinical trials for multiple myeloma are being conducted. So there's a government-sponsored website called clinicaltrials.gov, which you can search for myeloma clinical trials, and that'll point you to a medical center where there's a myeloma service. Cancer Research Institute also maintains web resources to identify clinical trials, and that can also direct you to a specialty center.
Okay, great. That's all the time we have for questions. Thank you very much for all of your questions. Just a reminder that our next webinar will be on May 28th, and the topic will be on reactivating your immune system to fight cancer. And We have a bunch more after that too. And as you may have heard, June is Cancer Immunotherapy Month. There's a lot going on, a lot to get involved with, and a lot of educational opportunities. It's going to be a lot of fun. So check out the URL. And also, if, as Dr. Cho alluded to, CRI does have a service that can help patients find clinical trials. It's called the Clinical Trial Finder, and the URL is on screen. There's a phone number that you can call and speak with a person who will help you sort through trials and find one that's appropriate for you. And finally, I'd just like to thank Dr. Cho and all of those of you who participated in today's webinar. Once again, this webinar will be available for viewing on our website within the week, and you can also check out past webinars there as well. And we hope that you'll take a moment at the end of the webinar to provide us feedback via the survey that you'll be directed to at the end.